Oh my goodness, I am so stressed out. I know absolutely nothing for this final, and it's in a week. I'm gonna fail this. What am I going to do?
Trav, it's not that hard, buddy. Especially when you run into the best cell bio coaches. What do you need to know?
I know absolutely nothing, and my final's in a week.
Well, we'll help you review. Let's go over something important like splicing.
So what is splicing?
Splicing is a modification made to RNA in eukaryotic cells after it is transcribed.
Before splicing, mRNA contains both non-coding sequences known as introns and coding sequences known as exons.
Introns are unnecessary, while exons are essential.
So how do you get rid of the unimportant sequences while keeping the important sequences?
Let us tell you.
Sequences within the RNA attack each other to cut out the introns, leaving only the exons, which then join together.
It isn't quite that simple though, Travis. There are numerous elements that contribute to the process of splicing.
Dan's right, as always. Elements within the RNA, called cis elements, help to splice. There are 3 cis elements: the 5' splice site, the 3' splice site, and branch point A.
The 5' splice site is at the end of the first exon.
And the 3' splice site is on the end of the second exon, leaving the intron in the middle of the 2 exons.
Oh yeah, I remember Dr. Ramos saying something about that. Isn't branch point A in the middle of the intron and attacks the 5' splice site?
That's exactly right. When branch point A attacks the 5' splice site, a phosphodiester bond is broken and a lariat structure forms.
What is a lariat structure? I think this would be easier to see this in action.
Well, it would be easy to explain with some props, maybe some drawing on the board.
Well, Travis, we couldn't find any props last night, but we can try some drawings on the whiteboard today.
First, let's review the cis elements. You've got the exon 1 with a 5' end, the 5' splice site, which is on exon 1, the intron in the middle with branch point A on it, the 3' splice site at the end of the exon 2 that's closest to the intron, and exon 2.
Now let's start with the process of splicing.
So you guys said something about lariat forming. What's up with that?
Yes, that's right, Travis. The lariat forms when branch point A attacks the 5' splice site, and it kind of looks like a last cell, as you'll see later on.
During this, the phosphodiester bond seen here is broken, and the OH group from branch point A is brought to the 5' splice site, as you can see here.
Then the 5' splice site attacks the 3' splice site, and a phosphodiester bond is broken here.
The hydroxide group that was just put on the 5' splice site is then transferred to branch point A again, and the exons are brought together.
So where does the intron go?
The introns will float off and eventually be degraded.
I've also heard of a spliceosome. So what does that do?
So the spliceosome is made of 4 RNA/protein complexes known as U1, U2, U4/6, and U5.
To break down the pieces of the spliceosome, U1 is what base pairs to the 5' splice site, and U2 binds to U1 and branch point A. This causes the RNA to bend into a U shape as seen here.
The last 2 pieces, U4/6 and U5, bind near the 5' splice site, and the spliceosome is complete.
In the intermediate stage of the formation of the lariat, U4 and U1 leave.
After branch point A attacks the 5' splice site, U5 holds the exon 1, and U2 and U6 stay on the intron.
And next, the intron is taken out and the exons are ligated together as seen here. And that's splicing.